Welcome to the last talk of the plant medicine track. The track will be closed with the participation of a very important researcher in this area that has set much of the knowledge that we all share about ayahuasca today. His name is Jordi Riba. He's from Spain, and he has a PhD in pharmacology. He leads the human neuropsychopharmacology research at the São Paulo Institute in Barcelona. And he has published more than 40 articles and chapters, and he's a great reference to all of us. Please welcome Jordi Riba.
So thank you, Bia, for the introduction. Thanks, everyone, for being here for this presentation. I'm going to talk about a psychoactive that it's not everybody's cup of tea, but I find especially interesting because it induces very profound modifications in the state of consciousness, and I hope I can show you some mechanisms that we've measured that might explain why the effects of Salvinorin-A do not quite fit the effects of the classical psychedelics like psilocybin, mescaline, ayahuasca, and so forth. So, first of all, to give you an introduction, Salvinorin-A has a structure, a chemical structure that's different from the usual alkaloids you've heard about in the course of this symposium. It doesn't have nitrogen on its structure. It's not a basic compound. It's what's called a terpene. It's obtained or present in the leaves of this plant, Salvia divinorum. And Salvia divinorum is used, or has been used traditionally by the same indigenous groups in Mexico that also use psilocybin mushrooms. That's the Mazatecs. So for several decades, it's been known that in addition to psilocybin mushrooms, for certain occasions, certain rituals, they use the leaves of this plant. So some years ago, it was discovered that it contains a family of terpenes, these non-nitrogenous compounds, and the one that's present in the largest concentrations is this substance here. It's called Salvinorin-A. So this was thought to be the active principle present in Salvia divinorum and potentially the one responsible for its psychotropic effects. So to give you some background, we were interested in Salvia divinorum in the mid-2000s because many people were experimenting with this drug. So we conducted a survey. We administered a series of questionnaires that we also used in the lab, and we gave them to people who spontaneously bought Salvia divinorum extracts and smoked them. And what we found, based on their responses, was that the profile of subjective effects induced by Salvia divinorum was somewhat similar to that induced by classical psychedelics. It has profound effects on perception. But in contrast to more classical drugs, it induces what's called a very intense derealization. The connection with external reality at certain doses can be completely lost. And the participants were also intensely impaired during the acute effects of the drug. So when you take ayahuasca, you're still able to interact with your surroundings. You may stand up, go to the bathroom, maybe dance, sing, whatever. With Salvia divinorum, at certain doses, if the dose is high enough, this is completely impossible. So then there was a first lab study in 2012, and they described the effects of Salvinorin-A when administered in vaporized form as being psychotomimetic and inducing dissociative effects, like the person lost touch with the physical body. And then the research group at Hopkins also replicated these findings. They described the effects of Salvia divinorum as having a partial overlap with classic hallucinogens, and also that the compound induced very intense dissociative effects. So classical psychedelics are known to interact with the serotonin 2A receptor. So the greater the affinity they have for this receptor, usually the more potent they are in animal models and also in humans. The more affinity, the lower the dose you have to take to get psychoactive effects from these compounds. But in a screening that was conducted in order to assess the affinity of Salvinorin-A for different receptor types, and this is what's shown on this graph on the right-hand side of the slide, it was seen that Salvinorin-A virtually only interacts with one receptor, and this is the kappa opioid receptor. So we have endogenous opioids. These endogenous opioids interact with mainly three types of receptors, the mu, the kappa, and the delta, and they found that Salvinorin-A was very selective for the kappa subtype. So the kappa receptor is involved in pain sensations, and it was also known to be responsible for what were the not so pleasant or dysphoric effects of classical opiates like morphine, buprenorphine, and similar compounds. But Salvinorin-A basically didn't interact with the mu or the delta receptor, only with the kappa. So in our lab, we conducted a first study, a dose-finding study. We published this in 2015, and we saw that at a high dose, you could get a complete blockade of external sensory perception. So we had participants in the lab. They inhaled various doses of Salvinorin-A, and when they inhaled 1 milligram of Salvinorin-A, they completely lost touch with external reality. They could be there with their eyes open. They wouldn't see the experimenters. They wouldn't see the room anymore. They wouldn't hear external sounds. So external sensory information was completely blocked, and they also lost the connection with their own body. This is what we call depersonalization, or a dissociative state. So we replicated these findings that other labs had already described. And we did, in this dose-finding study, we also measured the time course of effects. Here you can see the scores on a visual analog scale. In this visual analog scale, we asked the participant how intense are the effects. So we got a response from them. They had to rate the intensity of effects from 0 to 100. You can see here that at the low dose, you can get a response. The peak effects are attained between 1.5 to 2 minutes after inhaling Salvinorin-A, and the effects completely disappear after 10-15 minutes following the dosing. And at the 1 milligram dose, most participants were unable to give a response. They were unresponsive. So what we did was, if we didn't get a response from the participant, we rated the intensity ourselves as being 100. If you look at the highest dose, at 1 minute after dosing, the average is 100. So this means everyone lost contact with external reality. Again, that was published in that initial dose-finding study. And after that, we wanted to test whether indeed in humans the effects of Salvinorin-A are mediated by the opioid receptors. So we don't have a selective kappa antagonist that we can use in humans, but we have naltrexone, and naltrexone is a drug that you administer orally, and it is an unselective opioid receptor antagonist. It blocks the kappa, the delta, and the mu receptor. And in this study, we demonstrated that naltrexone was able to completely block the subjective effects of Salvinorin-A. We also administered ketanserin. Ketanserin is an antagonist of the serotonin 2A receptor, and serotonin did not reduce and the intensity of effects induced by Salvinorin-A. So here are some graphs for you to see. The violet bar shows the intensity you obtain when Salvinorin-A is administered alone. The blue bar on the left is the placebo. The yellow is the naltrexone, and the pale blue is the combination Salvinorin-A plus naltrexone. So what you see here is a very large decrease in the intensity of effects when you administer this combination, or it was rather a pre-treatment. We administered naltrexone 1 hour before participants inhaled Salvinorin-A. So you had a very large decrease in the intensity of effects, and the effects of the combination are not different from placebo in this subscale of the hallucinogen rating scale that measures perception. The same pattern of results we obtained for the subscale measuring cognition, and again, same results for the subscale of the HRS that measures affect. When we used ketanserin instead of naltrexone as a pretreatment, there were no differences between Salvinorin-A administered alone and the combination of Salvinorin-A plus ketanserin. This means that ketanserin did not have any effect on the intensity of the Salvinorin-A-induced subjective effects in any of these 3 scales I showed you earlier. So we did also an EEG study. So we placed electrodes on the scalp of the participants. We recorded spontaneous brain electrical activity. We used this technique in many studies with psychoactives. We've done this with ayahuasca many times and many other substances, psychostimulants, benzodiazepines, etc. So what I'm going to show you here is what we call the scalp topography. So these are statistical maps. The colors indicate statistically significant differences versus placebo, and the cold colors indicate decreases in power in a certain band, and the hot colors indicate increases versus placebo in another band. So the graph on the left-hand side shows you the effects induced by Salvinorin-A. That's the green graph. The red graph is the placebo. So after 1 minute after Salvinorin-A administration, so we measured we did 2 baseline assessments. There's no differences there between the green and the red lines. 1 minute after dosing, you see these large decreases in the alpha band. In this standard rhythm that you get when people are with eyes closed, and that have its main generators in the posterior cingulate cortex, in the medial aspects of the parietal cortex, where the key structure of the default mode network is. So what we saw after Salvinorin-A in this band, in the alpha band, was a very large decrease. You see that this is significant in almost all electrodes that we placed. It's 19 in total. But we found really an unexpected result with regard to the delta band. We had done studies before with ayahuasca, and with ayahuasca we found broadband reductions in power. So there were reductions in the energy associated with these spontaneous brain electrical oscillations in all frequency bands. But in this case, in Salvinorin-A, what we found here was a very marked increase. You see that the graph, the green line goes up, and the red colors or violet colors you see in this scalp topography indicates that there's a very large increase in the energy of this band, and you can see this all over the scalp. So here I'm subdividing, showing you results for Salvinorin-A, looking for what we call current density. So that was over the scalp. Here, what we did was localize which brain areas were contributing to these results we were seeing after Salvinorin-A, specifically in the alpha band. So this is subdivided in what's called the alpha-1 and the alpha-2 subbands. And as you can see, the areas that have their energy decreased are these regions in the medial parietal cortex. In this area that I told you is the key structure in the default mode network, and we also found decreases in areas involved in the processing of visual information. So this replicated results we had previously obtained for ayahuasca. This was a repeated dose administration study. We administered the first ayahuasca dose. You can see results in current density for the alpha band at 45 minutes, 60 minutes, 90 minutes, 120 minutes. So you see these reductions. This would be similar to what we obtained with Salvinorin-A. And again, when these effects eventually disappear 4 hours after dosing, if you give a second ayahuasca dose, the same pattern emerges again. So this would be a common finding with that. I mean, the results for Salvinorin-A would be in line with results obtained for a classical psychedelic such as ayahuasca. Other researchers using magnetoencephalography or MEG that measures the magnetic component of the electromagnetic signal have found also analogous results for the alpha band. So this is a study with psilocybin. It was published in 2013. And again, in the medial aspects of the parietal cortex, you see these large decreases. And also, more recently, this has been published for LSD. So Salvinorin-A has in common with classical serotonergic psychedelics. These marked decreased in the alpha band. This would be in common with them. But when we look at delta, these sharp increases we observed on the scalp, if we try to localize this on the brain surface, we see that there's a sharp increase in delta at 1 minute after dosing, and these delta increases are observed all over the cortex. So this is very unusual. This is not observed for the serotonergic psychedelics. This goes totally in the opposite direction. Normally, this delta rhythm is associated with a depression of the cortex. It has been observed as well with substances acting through an anticholinergic mechanism, like scopolamine, an antagonist of the muscarine receptors, induce this kind of changes. There's a large concentration of kappa receptors in the thalamus, so maybe these increases in delta are reflecting this inhibition of the thalamus, and maybe this is what is causing this loss of contact with external reality that we obtain with Salvinorin-A, and which is not at all a characteristic of the classical psychedelics. So, to summarize, Salvinorin-A at 1 milligram led to rapid and significant changes in the EEG power spectrum, coinciding with a peak of subjective effects. The main EEG effects were suppression of the alpha rhythm and increased slow delta activity. Similar to serotonergic psychedelics, alpha suppression was localized in the posterior brain regions, including the posterior cingulate cortex and visual areas. And delta increases were massive and were observed all over the cortex. So Salvinorin-A shares with serotonergic psychedelics the alpha suppression effect. However, it displays a typical enhancement of delta activity. And we postulate that Salvinorin-induced increases in delta may underlie the intense dissociation and loss of contact with external reality that differentiates Salvinorin-A from the classical serotonergic psychedelics. And with this summary, I conclude my presentation. Thanks for your attention.
Hello. Yeah, thank you very much for the presentation. Quick question: is it possible to reduce the delta wave activity with certain drugs or certain procedures? So could you isolate when you give the alpha Salvinorin that you only have the alpha increase, but can reduce the delta increase?
I didn't quite understand your
question.
Is
there a way that you can maybe pharmacologically inhibit that the delta stimulation is increasing?
Maybe if this is somehow being mediated by cholinergic neurons, like you're blocking their activity, because this is similar with what you get with drugs like scopolamine. Maybe if you administered some muscarinic agonist, maybe you could prevent this effect or block this effect.
Okay.
That would be my guess, but I really don't know.
Hello, and thank you. What's the subjective experience? Like, if they're not able to communicate, can they tell you afterwards where they went?
Oh yes, they went to a parallel reality.
Was it nice, or is it scary?
It was special. So we've met many people who have tried Salvinorin-A once and decided they didn't want to experience that again. But it was also interesting that in this those findings study we conducted initially, we recruited people who had extensive experience with psychedelics. Not all of them with Salvinorin-A, and we gave the doses in increasing order: 0.25, 0.5, and 1 milligram. So when they finished their participation, we asked them, if you were the researcher, which dose would you use in the subsequent study with 24 volunteers and EEG recordings, etc.? And they all said the high dose. So maybe it was strange, it was weird, etc. But I don't know, maybe they were sensation seekers, I don't know. All of them coincided. They said, 0.5, you get a glimpse of this other reality, but it's better to just go there and experience it.
Thank you.
Thank you for sharing this data. In 2005, I did EEG of Salvia, acute smoked inhalation, and found the same things. Alpha suppression, delta elevation. In addition, there's hypercoherence in high beta, and it's reversible. And it was just published as a conference paper. My colleagues didn't follow it up. I introduced it to Jorge Gonzalez in Mexico, who I think you cite there, and I turned him on to Salvia, and we did the EEG with him and all that. I have a poster here on DMT effects, EEG, and there's amazing overlap. Alpha is suppressed. There's another lab that has reported exactly the same thing. Gamma is elevated. It's nice to know that. So, yeah, Salvia is a whole different animal. But
And what do you see with, in the delta band with DMT?
Suppression.
Suppression as well.
Yeah,
Okay.
but sometimes hypercoherence in delta. Some subjects show that.
Okay.
Yeah, there's suppression of all the slow bands, and the elevation, you see it in the high beta.
I would like to see this conference
Come
paper
by
you mentioned.
The early one, yeah,
on
it's published.
Salvia, maybe you can email it
Yes,
to me.
of course, I can make that available to
Okay.
you. Thank you. I'm glad
Thanks.
it's been followed up. It's a fascinating compound.
Hi, thank you very much for the talk. So I feel like the most connected to Salvia. I've had the most profound experiences with Salvia. I've only smoked it. I've really wanted to experience the more traditional chewing. I also want to I'm getting transcranial subdermal implants, and a lot of these are going to be EEG contacts. Would you be interested in having a subject like me in a study to look at my brainwave patterns with an enhanced clarity given by a subdermal implant?
And why are you going to get these implants in the first place?
I think it's going to look fucking cool, and I want to shock my brain.
Okay, yeah, just call me. I'll be glad to. I'll be more than happy to do some intracranial recordings.
Thank you, very inspiring talk. You mentioned the increase in delta,
Yeah.
which we also observe in loss of consciousness due to sleep. For example, deep sleep, the delta waves increase in power. And as far as I understand, these subjects lose touch with external reality, but they...
Which population was that? I didn't hear that quite
So
quite
in
well.
general, I think there are quite a few publications showing increased delta in deep sleep, like
Okay,
loss
yeah,
of
sure,
consciousness.
sure, yeah, in deep sleep,
So, but in this experience, they lose touch with external reality, but they are still conscious, as far as I understand.
Yeah.
Do you know about the mechanisms, or would you speculate what is causing the difference between like being conscious of the self but not external reality versus being fully unconscious?
If you look at the raw recordings, at the raw EEG recordings during sleep, you can see these slow wave sleep, these delta rhythms, and they are huge. You can visually identify them. So you don't get these large increases in delta amplitude after Salvinorin-A. You cannot see this visually. It's after you do this power spectrum analysis that you see the increases. So there's already a difference there.
And a quick one, yeah?
Okay, after implant negotiations and parallel realities, I invite you all to go to the East Hall for the closing plenary. And thank
Okay.
you, Jordi Riba.
Thanks.